new viral infection will not take place, because the virus need a protein synthesis for their growth and then spread to the other cell. Okay. Now, if we have discussed about this anatomical and physiological barrier, there are some innate cellular response as well. So, talking about the innate cellular response, these are the phagocytic cell. We have the first line defense, that is anatomical and physiological barrier has been bridged. Then come to the, if they are coming to the tissue, now the body has to do something, and in such situation, innate cellular response, that is the phagocytic cell, that is the macrophage, that is monocyte, neutrophil and dendritic cell, they will come in the part to play the role in defense against the invading pathogen. They recognize pathogenesis via shared molecules that are not expressed on the host cell. So you have to understand, they have shared molecules that are not expressed on the host cell. So that is also important. There should be not attack your own host cell. So they are thinking, they are recognizing the shared molecule that is not present on the host cell but on the pathogenic organism. So that is should not be present on your own cell. So those molecules that are not present, that are different from our cell, they will attack that shared molecule and try to kill the pathogen. They are responsible for controlling the infection and sometimes even capable of eradicating them. Receptor that of the innate immune system are referred to as pattern recognized receptor, and they are pathogen associated molecular pattern, that is called PAMPs, and molecules that are shared by the pathogen of the same time, like bacterial LPS and N-formyl peptidide. So these are the bacterial structure, the molecules that are shared by the most of the bacteria. So LPS, all gram-negative bacteria have the lipopolysaccharide. This N-formyl peptidides, they present in the maximum bacteria. So they did not need to go for the specific, what you can say, specific antigen or specific epitope or specific receptor to kill this organism. They will recognize the sear. So all bacteria has lipopolysaccharide. So they will recognize that lipopolysaccharide that is not present in the human cell. So they can easily be recognized, and they are present in the all gram-negative bacteria. So they will target this bacterial lipopolysaccharide and act on all gram-negative bacteria and kill the infection. They can target the N-formyl peptidides that are present in the most of the bacteria, so that is not present in the human cell. So they'll target that, anything that comes and that have this N-formyl peptidides, they will attack, this cell will attack that and kill that organism. So in this way they are acting. Also they can kill the damage associated molecular pattern, that is called DAMPs. So there is another term, DAMPs, damage associated molecular pattern, released from the dying or damaged cell. These receptors are present intrinsically encoded in the germline and are not generated through the somatic recombination as the lymphocyte receptors have generated. So these DAMPs are actually not are generated initially, but when the cells are dying or diseased, then only they comes up. They are expressed. Initially they are not expressed. If they are expressed initially, they will be killed. So they are not expressed initially, but when they are dying or diseased, then they are generated, and then this will recognize by this innate cellular response, that is phagocytic cell, macrophage, neutrophil and dendritic cell, and that will kill. So they can kill the outer organism. They can also clear the associated damage or dying cell from our body as well. The innate response system can recognize only less than 1000 patterns and various pathogens, compared to the adaptive immune system, that is B anti cell, we can recognize over 1 billion of the specific sequence of the pathogen. So what we have to come to know, that this innate immune response, which have this anatomical barrier, physiological barrier, and as well as innate cellular response, whereas the cell comes up to play and act on this, as well as there is alternative complement pathway which are part of the innate immune system. Okay, so in this way, first they will try to prevent the organism to enter. If they enter inside, then the cell comes to take into action. How they recognize those pathogen? By this PAMP, that is pathogen associated molecular pattern, by which they recognize the all bacteria. Lipopolysaccharide is present in all bacteria, but not on the human cell. I mean mainly on the gram-negative bacteria. This N-formyl peptidase is present in the most of the bacteria, but not on the human cell. So if they found bacterial LPS, they go and attack that. They don't need to differentiate it is gram-positive, gram-negative, Streptococcus or E. coli. They did not need to be provide specific immunity. They are just recognize the common pattern, attack that and kill that. They are also responsible for clearing the damaged or dying cell from the body as well. They recognize only few patterns, that is less than 1000, and in comparison to the adaptive immune system, we can recognize multi-billions of the specific sequence of the pathogen. Let's end over here, then we'll continue our more concept or more chapter in the next lecture. Thank you.